Good afternoon, dear students, dear teachers, dear friends. It's really a privilege for me to introduce you our second Nobel Prize laureate speaker this afternoon. He is Professor Ciechanover, and he got the Nobel Prize in Chemistry in the year 2004, shared with Dr. Abraham Hershko and Irvine Rose. And the dedication of their Nobel Prize sounded for the discovery of ubiquitin-mediated protein degradation. Professor Ciechanover was born in Haifa, Israel in 1947. He got his MSc degree at the Hebrew University in Jerusalem, and he continued his MD, medical doctor degree, as well. He obtained it in 1973. And finally, he got a Doctor of Sciences in Biological Sciences at the Faculty of Medicine in the Technion, Israel Institute of Technology in Haifa. He got several very prestigious scientific awards, like the Albert Lasker Award in year 2000. This is more or less the equivalent, the American equivalent of the Nobel Prize. He got the Israel Prize in 2003, and as I mentioned, in 2004, the Nobel Prize in Chemistry. His work was focused, which was dedicated to the Nobel Prize project, was the discovery of the ubiquitination of target proteins. You already heard a very nice application of this area, since it developed as a very important general principle of getting rid of damaged or unwanted proteins from the cell. And if the system is not working properly, it may end up with very severe abnormalities like cancer or deposition of abnormally synthesized proteins like degenerative diseases. Here you can see just the event when Professor Ciechanover received his Nobel Prize in Stockholm, and he is member of several prestigious academies like the Israel National Academy of Sciences, the American Academy of Arts and Sciences, American Philosophical Society, and some other very prestigious scientific bodies. It's not the first time here with us in Szeged. 2 years ago, when we celebrated the 75th anniversary of our Nobel Prize winner, Professor Albert Szent-Györgyi, he was here with us. He had a very memorable lecture, very good time with our students, and even he participated in planting of the Tree of Sciences in the garden of the university. Today he is going to talk about, again, a very exciting area of biomedical research. He is going to talk about the revolution of personalized medicine. Are we going to cure all diseases, and at what price? Just please, Professor Ciechanover.
Hi, thank you very much. And I will try to run with you and start with the history of the ubiquitin system. And if we'll have time, we'll touch upon the personalized medicine revolution. And the reason is that I changed my mind in the last minute is because I heard this little wonderful talk about ubiquitination. And I want to tell you how the whole thing started and how it came upon. How it all, people are coming to the laboratory and feel that something big is missing. It's not like a tiny small question, but they feel that the whole floor in the building is missing and how they go about it. So we'll start with that, and then if we'll have time, and I want to be short because we are already a little bit late in time, talk to you about personalized medicine. What do I have to do with personalized medicine? I'm a physician. Actually, I never received a real formal scientific education. I got the Nobel Prize in Chemistry, but I never in my life taken any chemistry course, except in high school, and the little that they teach me in medical school, which you all know is not very serious. But it happens, you know, many of us, including you, are self-educated. You are pushing yourself according to the needs. If something is missing, if You are lacking a knowledge in some field, you are going and do it. You're just going and learn it. It's like in the lab. If you need to use mouse genetics, you go and learn mouse genetics. If you need to use fly genetics, you go and use fly genetics. You know, if you are saying, oh, I'm a biochemist, and all my life I grew up a biochemist, it will not take you far away. So during my life, I've undergone many transformations. I started with biochemistry, and now if you come to the lab, you will see that half of the people are doing mouse genetics, half are doing fly genetics, some are doing biochemistry. You just adapt yourself to the moving methodologies in science because you want to answer a question. And if the question needs utilization of new methodologies and new platforms that you didn't use before, then you go and adapt them. This is the approach, the right approach in order to make a progress. So let me take you to the ubiquitin system. And by the way, this is my city. You're sitting on a river and having very good freshwater fish that I enjoyed yesterday at Peter's house. But we are sitting on the sea, on the Mediterranean, not too far away from here. Tomorrow I'm flying back home. It's about 3.5 hours flight. So it's not too far away. And we have a beautiful city. This is the Baha'i garden. The Baha'i were in Iran. They were persecuted in Iran. And they migrated west via Iraq and Jordan, all the way and built a beautiful garden and temple. And underneath the temple, they buried their founder. So the founder of the center of the Baha'i religion is in our city, which is a little bit kind of strange because we are a Jewish state. They came from a Muslim country, but you know, life takes you to all kinds of unexpected directions. And the medical school is here. This is the medical school. So the city is built over a beautiful mountain of the Carmel. This is the very east coast of the Mediterranean, and the medical school is here, and I have a beautiful view from my window directly to the sea. So when people in the lab make me nervous, I look to the wonderful sea and I cool down. Okay, so the whole thing started from this experiment. It's not a very complicated experiment. Even non-scientists can do it, but they can do only half an experiment. You can take an egg and break it and make a fried egg out of it. But you cannot take the fried egg and put it back to the shell. Nobody in the world can do the reverse. It's a one-direction experiment. You can take meat. This used to be an Hungarian sausage, a good Hungarian sausage, now not anymore. And you leave it in the room temperature and something happens to it. It either take mold or become greenish or brownish and stinking. And so what's common between the two experiments? What's common between the egg and the meat? Temperature. High temperature. In the first case, it's really high temperature, but it took only a few minutes. You put the egg on the pan, and within a few minutes you have a fried egg, and you enjoy it and you eat it. This case is a little bit slower because it's in room temperature. But the lesson from these two experiments, proteins hate high temperature. They cannot survive in high temperature. The place of proteins, if you want to keep them for a long time, is in the deep freezer. If you go to the butcher, you bring a piece of fresh meat home, and you want to keep it for a long time, either you cook it immediately, or you put it in the freezer. Now think about our body. Our body is at a very high temperature, much higher than the outer temperature. We don't feel it. And whenever the temperature outside rises to the temperature of the body, to 37 centigrade, we say, wow, it's awfully hot. But we don't feel our body temperature because of different mechanisms. But nevertheless, our body temperature is 37 degrees. And 37 degrees is, as I said, and you know it already, and even lower than that, is deleterious to proteins. So our proteins in the body are not any different than the proteins in this piece of meat, right? Well, we are thinking of steak as being taken from the cow, but we have steak here. We don't want to think about it because we are not cannibals. But the muscles in my thigh and in my arm are basically a piece of steak, isn't it? And this piece of steak doesn't like to be at 37 degrees. But it has to be at 37 degrees because this is the optimal reaction that was picked up by evolution in order to catalyze optimally all the biochemical reaction. I cannot be in the freezer because I'll die in no time. I will not be able to generate energy to burn glucose, to burn amino acids and lipids. So 37 degrees was chosen, selected along the years as the exact temperature in which, on one hand, biochemical reactions are running optimally, but on the other hand, we are paying a price for living at 37 degrees, and the price is constant denaturation and misfolding of proteins. So while you are sitting here and listen to me, and while I'm talking to you, the proteins of all of us are getting denatured and in unbelievable pace. So the evolution and the body had to find an answer to this problem. So here you have a problem in biology. How do we live in 37 degrees? And there are several answers to it. Actually, we just heard on the DNA repair mechanism, the body had evolved numerous repair mechanisms. One of the repair mechanisms for proteins are chaperones, HSP70 and HSP90 and all these chaperones that are basically refoldases. They are refolding the proteins with an investment of energy. But not every protein can be refolded. The egg protein that was put in 400 degrees in boiling oil cannot be refolded. And many of our proteins cannot be refolded, and the only way to handle the problem is to remove them and to replace them with new ones. So here already, theoretically, we are just joking about eggs and Hungarian sausages. We are coming to a major problem in biology. So the body had to evolve a very sophisticated mechanism in order to remove protein that cannot be refolded by chaperones and to replace them with new ones. And we discovered this mechanism, and that's the ubiquitin system that you heard already. I will not go into details, but I will just take you and lead you via the thinking and the discovery process of what do you do. So the problem in biology is that you have a problem, but that's not enough. Now comes the hard part. You have to design a way to solve the problem. You can say, cancer is a problem. So what? How do we solve it? Say, aging is a problem. We don't want to age. We want to be young until we are 250 years old. But so asking a problem, asking a question is the first stage. But then you have to pull your sleeves, go to the laboratory like Ada did for 30 years, and solve the problem. Because the bigger the problem, the longer the time and the more complex is the solution. So there is a problem here, as you can see, and we need to solve it. And the problem is denaturation of proteins. Now, if I give you the numbers are staggering. Every day, we are degrading, destroying about almost 10% of our proteins, something between 6 to 9%, depends on different metabolic conditions. So you can imagine that along 1 or 2 months, there are proteins that are living very long, like hemoglobin, that is living for 120 days. There are proteins that are living shorter than that. But you can imagine that along the time, 1 or 2 months, we are replacing all our proteins. So now, what is today, the 26th of May? I'm made of completely different proteins that I was made on the 26th of March, or the 26th of February. So I'm a new man. How nice. But there is a problem there still. First of all, how is it that I changed all my hardware? That my hardware was changed, but my software remained the same. Or I think so. So my memory is with me, except that I forgot half of the things that I remembered a few months ago. My passion, intelligence, I remember my address at home. I know tomorrow to go back home. I know to tell the driver in the airport where to take me. Or I hope so. So think about it. It's a major problem. How is it if you buy a new computer, the new computer can be identical to the old computer, but you have to take the software and move it from one computer to another. I am changing my computer all the time, my body all the time, and the software remains the same. It's not a simple problem. It's a very complicated problem because we are exchanging ourselves all the time. We are in a very dynamic state. Within 2 or 3 months, not a single molecule of protein that had been with you 3 months ago is with you today. Then comes the question of aging. How is it that I'm aging? Why is it that I'm aging? Why is it that at the old age I get cancer? That the body succumbs? Why is it that the diseases that Ada talked about, pneumonia, that I can cure easily, a 15-year-old boy, old patients are dying of it, and I cannot do anything? It's not because the machinery is not working. It's because that other systems are compromised. The immune system and other systems are compromised, including also the ability to replace proteins. You can imagine that because we are living so long, you don't have to die from a car accident. You know, if a car runs over you, you die immediately. Basically what we are doing, we are dying along decades. So a little deviation of the system that we discovered, let's say that the system now can remove only 99% of the proteins that it should remove, and 1% remains. So it's 1% today, and 1% tomorrow, and 1% along the month, and 1% along the year, and gradually I accumulate the damage. You talked about Parkinson's disease. In order to get Parkinson's disease, we need to lose 80% of the cells in the substantia nigra par compacta, 80%. And this takes decades. And only once we are losing 80%, we start to see the first tremor, the first clinical symptom. So we have a big reserve. But the disease does not start when we see it. The disease started many years ago. The same for heart attack. People say, oh, until yesterday he was jogging along the river here in Szeged, and then all of a sudden he died of heart attack. It took him 4 decades to die because he accumulated gradually, degenerated the coronary artery and accumulated cholesterol until the very last block came. So what we are talking about gradual compromise of quality control mechanisms. Think about it. You know, now we are trying to define what is to die. Dying from aging and aging at all. You know, the fact that I'm now not as young as I used to be and my system know it, is gradual, slow compromise of quality control mechanisms. And the ubiquitin system is one quality control mechanism. And you don't need to shut it down in order to die. It's enough that it will gradually lose its function by 1%, 1 little percent. But this 1 little percent will run along 10 years, and here you are with Alzheimer and Parkinson and ALS and all the other degenerative diseases of the brain and many other. Cancer is a degenerative disease. It's a degeneration of the quality control mechanism that controls cell division rate. We are losing the ability to control the Swiss clock of our body. So I'm taking you a little bit through philosophy because I think that it's important to think of science in a global way rather than in experimental terms. Once you have the idea, then you can go to do the experiments. Okay. So destruction in the body is not necessarily, as you see, people thought about destruction for many years as kind of neglected area. Why we have to destroy? There is the beautiful ribosome and the DNA and the RNA, and we are making proteins, and destruction was kind of neglected. Now I convinced you already that destruction of harmful protein, misfolded protein, is essential for life. but it's essential also for development and for prevention of cancer. Think about apoptosis of the interdigital space. You know, the fingers are not generated genetically, they are generated fused. And gradually we are destroying the interdigital space. So destruction is important also for development. And most importantly, daily we are destroying billions of cells that have undergone mutation, DNA damage that could not be repaired. And all these cells have the potential to become cancerous cells. So we have to destroy them, again by an apoptotic mechanism. So, you know, destruction and construction in the body are acting in balance. But scientists are not acting in balance, because scientists are like fashion designers. They are after fashion, so which acted actually at our favor. Because in the 50s and 60s and until actually almost the 90s, all the scientists were busy with the central dogma of biology, with DNA, genetic code, RNA. Nobody cared about destruction. And the field was empty. Apoptosis and all these fields are relatively new. And the same for the ubiquitin system, for the need to destroy proteins, which led us in Israel, which is a tiny small country, smaller in surface area than Hungary, much smaller than Hungary. Population, we are a little smaller, we are 8 million, you are 10 million. To identify a niche. So being in science is not just, you know, being in science, being good, but also identifying a niche in which other people think that you are stupid, that you are going to something that is unimportant, and identifying a niche that nobody cares about. So we purposefully identified the niche, because if we would have run to DNA at that time, we would have been lost, losing against all the American sharks. When you're in a small place, in a small town of 200,000 people, our city is 200,000 people, and you are in a poor country, you need to think also strategically and to ambush your enemy, to prepare some surprises for them. And once they wake up, it's too late. It's as the French people say, it's a fait accompli. That's it. Okay. So the first people that thought about protein degradation, one of the first people was Rudolf Schenheimer in Germany, and he used to give labeled amino acids to rats. And once he gave them to rats, he found that the amino acids are incorporated into proteins and then come out of proteins. So he was the first to identify a dynamic state of the body constituents. And he committed suicide at an early age, and it says that his work helped establish that the living body is a chemical laboratory. The body itself is a laboratory where complex transformation of matter are taking place all the time. So he basically identified the fact that we are dynamic, that we are not static. And people did think that we are static, that we are being born, we are eating initially the good milk of our mother, and then the good food that our mothers buy in the supermarket, or wherever she buys it, and we grow up, and all what we are doing, we are accumulating, but we never lose. The idea is that we are synthesizing along with destruction in a dynamic way is a relatively new idea. People didn't know about it. It is a new idea. And Rudolf Schenheimer in the late 30s and early 40s introduced this idea. He was Jewish, living in Germany. Needless to say that the German kicked him out. He went to the United States and continued the work in the United States. I just wanted to point out to you historically that all this movement of Jews from Europe, including of Einstein that was shown earlier by Ada, who was also Jewish, is called Hitler's gift. This is the gift that Hitler gave to the West. By kicking out all these smart people, physicists, chemists, biologists, and by that enable the West to build up its modern science. American science, modern American science, and modern English science even. Kendrew and Perutz was a Jew that escaped from Austria. And many of them left Europe, emptied Europe from its intelligentsia, and went to the West. But I will not stop here. And then came when people realized that proteins are degraded, they looked for the mechanism. So initially it was Rudolf Schenheimer, and now I want to tell you how you get into the problem. Now, first, as I said, there is a problem, but now you need to say, okay, then what do I do in the lab next? I'm coming to the lab in the morning, what do I do? Where do I start? So Rudolf Schenheimer did simple experiments to find that proteins are degraded by giving labeled amino acids to rats and finding that it's coming out. So proteins are dynamic. They are being synthesized and degraded, even in the young rat. Now came Christian de Dive, and in the early 50s, he just died a year and a half ago, fantastic Belgian scientist that worked both in Leuven in Belgium and in the Rockefeller Institute in New York. And he discovered this organelle, the lysosome. So the lysosome is an organelle inside the cell that digests cellular protein. It has proteases inside. So inside the lysosome there are proteases, and it digests cellular protein. How? It digests them by taking off droplets. You see here there is a droplet here. So they pinch off a droplet, they make it a vacuole, the vacuole open, and all the proteins are degraded. So everybody was happy. Initially they didn't believe that proteins are degraded, and Schenheimer came and told them that proteins are degraded. Then they asked a question about the mechanism, and Christian de Dive came and showed them the mechanism. The mechanism is the lysosome. So then what else is left to be done? All the questions are answered. From static life, we moved into dynamic life. From no understanding of mechanism, we have a mechanism. Christian de Dive, by the way, along with George Palade and Porter, Keith Porter got the Nobel Prize for different discoveries in cell biology. They shared the Nobel Prize for Medicine or Physiology in '75. So what is there? This is there. People discovered gradually, and here we are jumping already to the mid-70s, that different proteins have different half-lifetimes. So each protein has its own stability. Some proteins are very quick. They are generated and degraded, generated and degraded. And some proteins are very slow, like hemoglobin. They are synthesized and live for 120 days. And you see the variety is huge. Some proteins are living in the cell 0.1 hour, 0.2 hours, 12 minutes. Some proteins are living 15 hours. Almost 100-fold difference and even more. This cannot be the mechanism, because remember, the lysosome acts by pinching off droplets. Now all the proteins that are in the cytosol are represented in this little droplet. Think about if I take a nylon bag now and I sample air here, the composition of the air will be 79% nitrogen, 21% oxygen, and little bit, 0.12% carbon dioxide, and little noble gases. If I take the same nylon bag and go over there, it will be the same, more or less. So the same is for proteins. How can it be that some proteins are very short-lived and some proteins are very long-lived if they are all included in this little vesicle that goes in and destroys? And not only that, people discovered, for example, that some proteins are very stable, cyclins, the regulators of cell cycle. They are stable all along the cell cycle, and in 5 minutes, when the cell goes into mitosis, they are degraded. How can it be? What is it in mitosis that makes certain proteins degraded? So people realize that there must be a more complex, specific mechanism that can degrade proteins specifically, and also abnormal proteins. Why abnormal proteins? Think about the cell. In the cell, there are, let's say, 1,000 molecules of the same type. Okay, hexokinase, glucokinase, that's how that. And one now became denatured because of heat. We want to take this one and degrade it. But all the other 999 we want to keep intact. How do we do it? This cannot do it, because everything is inside. We need to have very unique pair of spectacles that will pick up with forceps only one molecule that is denatured because of heat or because of oxygen or because of mutation. degrade it, and all the others should be spared. You need a highly specific mechanism. So see how processes of thinking are coming in biology. Initially there is a problem, you say proteins must be degraded. The steak, 37 degrees, somebody must take care of it. Then they have to show that there is indeed dynamism, and Rudolf Schenheimer showed it by his labeling experiments. Then came Christian de Dive with one mechanism, and for many years people believed it, but then they started to ask questions because more knowledge came. It's not that Christian de Dive was naive, he was not naive at all, but for his time, this was sufficient, because people didn't have half-lifetime measurements of individual proteins. People didn't know anything about cell cycle and cycling. Once more knowledge comes, more problems are opened, and people say, no, the lysosome cannot be. Something else must be there. And we came into this problem exactly when this question started to rise. Luckily, there were not too many people to ask this question, because everybody was busy with DNA, and along with my mentor, Avram Hershko, who is half Hungarian, and I think he visited Szeged, and there is a bronze bust of him in the university. I was a graduate student. We started to work on the system, on a putative system. It was not a system, nothing existed. We hypothesized that there is a system, and we started to look for it. And how do you look for it? You put an assay. I will not go into it. We started with red blood cells, with reticulocytes. We opened them up. We put a proteolytic assay, and we started to clean it. You say, there must be something. Why we started with red blood cell? Again, now we are starting to go into the details. And I will not go into details, but I just show you how you take the thinking and bring it to the laboratory. What you do is you ask yourself, okay, now I want to do an experiment. So the first experiment you ask yourself, what is the biological system that I pick up? And we picked up this cell. Why? Because this cell doesn't have a lysosome. It's an already terminally differentiated cell, no lysosome. So we knew that anything that we shall find will be new. Because if there is a lysosome, then you can get confused. But if there is no lysosome, and the cells still degrade protein, then it must be something else. So you already start with an assumption or with a system that makes your life easy. So picking up your experimental system is critically important in any experiment that you do. You need to pick up a system that will give you the right answer. Because otherwise, you may run into an artifact. We could take a cell, and everything that we would have shown would have been lysosomal. So what you do, you discover something that was discovered 50 years ago. This is not science. So we started with a cell that we knew doesn't have a lysosome, and very quickly we discovered proteolytic activity, and then we started to go to columns and fractionate it, and we purified ubiquitin, and we found ubiquitination, and we found E1 and E2 and E3, all the ubiquitination machinery that was mentioned here already. And it took us about 7 years to put the principles of the system together. I will not go into details, I just show you how the system looks now. And the system looks like that. We have a protein. This is a protein that has to be degraded. Target. This is the target protein. It is bound to a ubiquitin ligase. I'll immediately explain to you. This is a protein that binds to it ubiquitin. And ubiquitin, according to the Nobel Committee, is the kiss of death. Once ubiquitin is bound to you, you are dead, or almost dead. Then comes, here is ubiquitin, a small molecule. It is activated by E1. I will not go into the mechanisms. We worked out all the molecular and biochemical mechanisms. It's going from another enzyme, from E1 to E2, and then it goes to the substrate. And it goes to the substrate multiple times, and it marks it to its death. But the protein is not dead yet. It only had the mark on the back. And now comes the scissors, which is called the 26S proteasome. And the scissors recognizes ubiquitin. It doesn't recognize the substrate. So this is the sparing mechanism. If you don't have ubiquitin on your back, you are okay. Only proteins that are denatured and misfolded get this kiss of death. And once you are kissed to your death, you are dead. And then the protein is being opened and cut into pieces. So you need this glue, ubiquitin, to bind you to the scissors. And we discovered all the mechanisms and everything involved. Now, in 2000, now we are running very close now, it's only 14 years ago, the human genome was discovered. Because we did all our work, not by genetics even, we did it by biochemistry, pure biochemistry. And the human genome that was discovered told us that the system that we discovered has close to 2,000 proteins. In the human genome, there are about 22,000 proteins, 22,000 genes. 2,000 of these genes, 10% almost, belong to the family of the ubiquitin system. Nobody could have imagined before that a garbaging mechanism will be so complex, but now we know that it's not only garbaging mechanism. It is involved in many processes in the cell, including DNA repair, endocytosis, exocytosis, cell cycle, and so on. So ubiquitination serves not only to mark proteins for degradation, but there are different forms of ubiquitination. I really don't have the time to go with you into it, but you can see here there are numerous ways to ubiquitinate protein. And each way signals the protein for something else. Some for DNA repair, some for cell division, some for degradation. So you see ubiquitin can do chains like this and chains like this and linear chains and monoubiquitin and short chain and long chain. It can do all kinds of things, and each type of a chain serves something else. Let me just devote the last 5 minutes to show you that we are not in theory, we are in real practice, and the real practice is obviously medicine. So now we are coming back. Medicine is drugs, and drugs are aimed at patients, sick patients. There are already several very successful drugs. I will show you only one, based on the ubiquitin system, and I'll show you just one drug, which is an inhibitor of the scissors. You remember, once your protein is ubiquitinated, it goes to the scissor to be cut. So it's an inhibitor of the scissors. I don't have the time to go into it, but I want just to show you the principle beforehand. There are many drugs in the ubiquitin system, and there are many diseases that are related to the ubiquitin system. I mentioned some of them. For example, here you have cases of Alzheimer's disease. This is early onset Alzheimer, and you can see here immunohistochemical staining for ubiquitinated proteins. So proteins are ubiquitinated in the brain, but the scissors cannot take them. So they accumulate. So we see this in many cases of early onset juvenile type of Alzheimer's disease. But I will not go into it. I want to go to cancer. Now you can imagine that cancer is a disease where cells are dividing rapidly, right, to make a tumor. And normally the cell has to divide, let's say, in 24 hours. So you can imagine that the car, you can go, instead of thinking of the cell, you can imagine a car. And a car has two major control mechanisms. It has the gas that you accelerate, and it has the brakes that you slow down. And in between, you are driving. Obviously, you have a steering wheel. But if you have a straight line, a straight street, you don't need the steering wheel. You can control the car by the brake or by the gas. Now, when the cell divide, you push the gas, right? So if you want to imagine the metaphor, the analogy, the gas is the division. If the cells stop dividing, it's like the brake. Now you can imagine that if the cell somehow increases the degradation of a tumor suppressor, and a tumor suppressor is the brake. Right now I'm going back from the car to a tumor suppressor like P53 is the brake. It's a quality control protein. If the cell will somehow increase the degradation, the car will remain with no brake. If the car will remain with no brake, it will run and accelerate, and this will be cancer. So here is cancer. On the other hand, if the cell will not degrade growth promoting factors like the EGF receptor, and they will accumulate, again you will have excess of gas. So either increase degradation of the brake or decrease degradation of the gas pedal of the proteins that are the gas of the cell will bring cancer. So here you can see already, and there are many cases here, here you can see already that imbalance between the brakes and the gas in the cell will lead to cancer. So an inhibitor was developed, and here is the inhibitor. It's an inhibitor of the scissors, as I told you. Oh, sorry for that. This is the scissors, the proteasome. It has active site. It's a protease. And here an inhibitor was developed. It's a tripeptide. It's a boronic acid derivative. I will not go into it. And it's make magic on patients with a specific leukemia, which is called multiple myeloma. It's a B-cell leukemia. It's a leukemia of the B cells that make antibodies. And people are dying of it very quickly, relatively, in 2 years. And this drug changed completely their lives. And I can show you a patient with multiple myeloma. Here is one patient. And as I said, this is multiplication, uncontrolled multiplication of B cells that make antibodies. Here you can see the antibody is coming up very quickly because the cells are dividing. We started chemotherapy, little response, coming back, and here started a treatment with the inhibitor. The disease goes away completely. You can see the bone marrow, which is the factory of the B cells, and you can see here the bone marrow is completely homogeneous, full of malignant cells, 41% malignant cells. After treatment, less than 1%. All the normal cells are coming back, and the malignant cells are gone. And you can see here a cousin disease, a similar disease, which is called Hodgkin lymphoma or non-Hodgkin lymphoma. You see here a patient with non-Hodgkin lymphoma. You can see the tumor here. This is the chest. You see here, this is the bone, the front bone. This is the vertebra. This is the shoulders. These are the big vessels of the heart. And you can see here a tumor sitting. This is the lung, the right lung. And following treatment of these lymph cells, the tumor is gone. So let me conclude here because I'm running out of time and just give you the very bottom line. When we started the work, we never thought of drugs. We never thought of cancer. We never thought of anything. We were asking a question. We were trying to identify a niche, a big niche in biology that was still open. And gradually, and we started to read the literature. So we said, aha, there is a problem with the steak at 37 degrees. And then we went to Ciechanover and we found, yeah, there is indeed a problem and the body can degrade it. Then we went to Christian de Div and we looked at the lysosome and we said, the lysosome is not the answer. There must be something else. And only then we went to the laboratory to start to look for the something else. So it was kind of an unbiased search or an unbiased lookup for a system that was not existing at all, that we hypothesized based on assumptions in the literature that it exists. So we worked and worked and worked and found the system. And then other people came and said, wow, this is interesting. Maybe if defects in the system lead to disease, we shall develop drugs. So you see how science is rolling. We have nothing to do with the drug. We did not touch the drug. But that's the way that science is moving. From a very primitive question all the way to the lab and then to help the patient. So you see here, I was lucky enough during my lifetime, well, it was quite long. We started the journey in the mid-70s. So we are already 40 years into the race. But you see, 40 years, still okay, I'm alive and still running around. So in 40 years, you start from zero. And then you are putting into science a whole system that you are the one that discovered it. And the system is not theoretical system. It's very practical because it brings help to lots of patients. So I think that I don't have time for the second one. Maybe Peter will invite me once more for the next time and then we'll do it. So thank you very much.